Hello, everyone. Thank you so much for joining us for today's webinar. This webinar is put on by NanoImaging Services. We are a US-based Cryo-EM CRO focused on structural biology and nanoparticle characterization services. 1 year ago, we acquired Proteos, a protein production CRO based out of Kalamazoo, Michigan. So we now offer the full gene-to-structure workflow, as well as standalone protein production. Today, we'll be focused on epitope mapping of structurally heterogeneous samples using the SARS-CoV-2 spike protein as a case study. This is work that was done in collaboration with the Phillips Lab at UCSF. During the presentation, we encourage you to post any questions you have in our Q&A toolbar. Following the talk, we will have a live Q&A session, and we'll answer all of your questions then. Now, without further ado, it is my pleasure to introduce today's speaker, Dr. Kenny Kang. Kenny has been with NIS since 2021, where he serves as a senior scientist and project manager. He is an accomplished structural biologist who completed his PhD and postdoctoral training at MIT, specializing in cryo-EM techniques to understand biological macromolecules. Prior to joining NIS, Kenny was a visiting lecturer of chemistry at Wellesley College. Kenny brings deep expertise in structural biology and cryo-EM to his work at NIS. So please join me in welcoming Kenny Kang. Kenny, over to you.
Thanks, Sarah, for the introduction. Hi, as Sarah mentioned, I'm Kenny Kang. I'm a senior scientist here at NIS on the single particle analysis team. Thanks, everyone, for joining today's webinar. We're excited to finally present this story that we've been working on, where we solved a series of different Fabs bound to different variants of SARS-CoV-2 spike protein to try to structurally characterize antibody evolution using Cryo-EM. As Sarah mentioned, this project was born out of a collaboration with Angela Phillips's lab at UCSF. The Phillips lab is broadly interested in studying the molecular constraints on protein evolution and coevolution at the host virus interface. They use flu and SARS-CoV-2 as model systems to do. So towards this goal of studying these molecular constraints, the lab has developed tools to generate large, on the order of 10 to the fifth, combinatorial libraries of mutational sequence space to study how trade-offs between different biophysical properties like stability, folding, and function affect the evolution of these antibodies or viral proteins. So essentially, how these different biophysical properties affect the mutational landscape as an ancestor evolves and acquires mutations towards a functionally evolved variant. So for this specific project, the Phillips lab was interested in getting the structures of two different antibodies, what we'll call OMI germline or OMI GL and OMI 32. And these are two antibodies that bind the receptor-binding domain, or RBD, of SARS-CoV-2 spike. So OMI 32 is a broadly neutralizing antibody that was isolated from a breakthrough patient that was reinfected after vaccination. And this OMI 32 binds different variants of spike quite well, such as Omicron BA1 and BA4. And then based on the sequence of this OMI 32 antibody, the Phillips lab inferred the ancestral antibody sequence from which it likely evolved and generated that antibody in the form of OMI GL or OMI germline. And so the overall goal here was to try to structurally characterize these two antibodies, which are the evolutionary endpoints, the germline ancestral antibody and the evolved variant OMI 32, bound to these different variants of spike from Omicron BA1 and BA4. So the samples that we received from the Phillips lab are these four proteins. We received BA1 spike protein, BA4, and then the two antibodies, OMI germline and OMI 32. And our goal was to get structures of three of these pairs, BA1 plus OMI GL, BA4 plus OMI GL, and BA4 plus OMI 32. The fourth pair, BA1 plus OMI 32, the structure of that was previously solved using crystallography. And so we had omitted it from our series of structures. And Around the time we kickstarted this collaboration, we were also internally interested in whether we should start using this anti-LC kappa VHH for other client projects as well, in case it could be beneficial. So this LC kappa VHH binds to the hinge region of the light chain of kappa antibodies. It can act as a small mass enhancer, but also can help to stabilize the C terminus of the Fab, which can tend to be flexible across that hinge. And so we sought to get structures of each of these pairs in the presence and absence of this LC kappa VHH. So on our end, this work was largely spearheaded by myself and my colleague, Claudio Catalano, of course, with a lot of contributions from our lab operations team as well when it comes to grid preparation and imaging. For each of these samples, we collected all of our data sets on a Glacios 200 kV microscope, collecting somewhere in the range of 3,000 to 5,000 images for each data set. And it turned out for this sample, it was most beneficial to collect these data in intermediate or medium ice thickness. When we went a little too thin in the ice, the complex tended to fall apart. And then if the ice was too thick, the common thick ice problems like overlapping particles and lower contrast is what we faced. That said, we got a little lucky as we started this project that aside from nailing down the appropriate particle concentration and ice thickness, there wasn't too much additional grid optimization that was necessary. So we didn't face any challenges with preferred orientation or anything like that. So we didn't need to do any additive screens in this case. So a little bit of luck there. So starting with just looking at some representative 2D class averages from one representative sample pair, so BA1 and OMI germline. On the top, I'm showing some 2D classes of the sample prepared without the LC kappa VHH, and on the bottom, prepared with the LC kappa VHH. And a couple things that stood out. Overall, the 2D class averages looked pretty promising from the start. Different particle views appear present, and we see higher resolution features. If we take a closer look at the 2D class averages, where we see those highest resolution features, like the secondary structure, appear to be mostly in the spike protein itself. And then if we go out and look towards the fabs, the density starts to get a little hazier, a little blurrier, which we took to mean that there's some flexibility or conformational heterogeneity at that RBD fab binding interface or around that area. We also noticed that different 2D classes, we saw different numbers of fabs bound as well. So some 3, some 2, some fewer. And so in addition to that conformational flexibility, there appeared to be some differences in occupancy as well, or some degree of compositional heterogeneity in the samples. As we started to do some 3D processing and classification, our initial observations mirrored what we saw in the 2D class averages. So on the left, I'm showing some 3D classification results of the BA1 and OMI-GL without the VHH added, and nothing too surprising based on the 2D classes. We saw different numbers of fabs bound to certain classes. Sometimes the fab was bound to an RBD that was in the up conformation, sometimes bound to an RBD that was in the down conformation, which is something that had been observed before many times. I think one of the more interesting observations at this stage was when comparing it to the sample prepared with the LC kappa VHH. In that sample, it looked like most or all the fabs were bound to RBD that's in the up conformation. And so our current thinking is that it's possible that the extra sort of bulk contributed by the VHH bound to the fab maybe precludes the fab or at least multiple fabs from binding to the RBD when it's in the down conformation or pushing it towards being bound to the up. And so, yeah, that was one interesting difference between the two. In either case, at this stage of processing, we did end up taking whichever class showed best and strongest density for that fab RBD region forward. We weren't necessarily too concerned with trying to carefully quantify each of these different states at this stage, but one strength of Cryo-EM, I think, is if that is a question that you're interested in, you can extract that kind of information from a single data set. So the next slide, I'm just going to take this one example of the BA1 OMI-GL with the VHH forward, but I will say that the general pipeline we did apply to the rest of the samples as well. And I also forgot to mention that all of our processing for these data sets were performed in CryoSpark. So after some 2D classification and 3D classification to clean up the data sets initially, for this one sample set, so we took about 640,000 particles into iterative rounds of heterogeneous refinement and 3D variability analysis to try to hone in on a better subset of particles that have the best possible density for that Fab RBD area. That resulted in about 100,000 particles at this stage, getting us fairly close to where we needed to be. But in the end, for all these samples, we did have to go through some different rounds of local refinement, playing around with different masks, different local refinement parameters to sort of get us across that finish line. But for each of these samples, we were successfully able to do so. And each of these data sets, the BA1 plus OMI-GL, BA4 plus OMI-GL, and BA4 plus OMI-32, processed to somewhere in the 3.2 to 3.3 or 3.4 angstrom resolution with and without the VHH. And at this resolution, we could fairly clearly make out the density for most of the side chains at the antibody-antigen interface and then build into our experimental maps. So at this stage, we had sort of accomplished what we had set out to do initially with our collaboration, solve this series of structures, and we were able to do it fairly quickly. But then the Philips lab was also interested if we could also get the structures of the Fabs by themselves, so not complexed to the antigen. The Fab around 50 kilodaltons is on the sort of smaller side, but I think we were especially encouraged by what we were seeing when we added the VHH to the Fabs. So not only was it a small mass enhancer itself, but it was stabilizing this C-terminal region of the Fab. And I'm not showing the maps here, but the maps generated without this VHH, the density for this upper region of the Fabs are less well defined. And so I think with that information in hand, we did push forward to try to get the structure of the Fabs alone. And for that, given the size, we did transition our data collection over to the Krios 300 kV microscope. But in the end, we were able to generate the structures of the Fabs alone in complex of the VHH to about 3.2 angstrom resolution. And that allowed us to make some pretty interesting observations comparing these structures to the antibodies bound to the spike protein. So here, I'm showing an overlay of 4 different structures. So in light pink, or the lighter shade of pink, is the germline antibody bound to BA4. And then the lighter shade of blue, the OMI-32 antibody bound to BA4. And then in the darker shades of each of the colors, the Fab alone of the germline and OMI-32. And when we align all these, we see that for OMI-32, the CDR2 loops in both the light chain and the heavy chain, the conformation that the loop is in is the same conformation that it's in when it's bound to the antigen. In contrast, for the OMI-GL shown in pink, and the insets are of just the Fabs by themselves. So the OMI-GL shown in pink, the loop conformation shown here is when it's unbound, not bound to the antigen. And then when it's bound, it adopts the same conformation that the blue loop is in, or that the OMI-32 loop is already in. So from this, it seems like for OMI-32, the evolved antibody, the CDR2 loops are sort of pre-configured prior to antigen binding. And then for OMI-GL, these CDR2 loops appear to need to go through some conformational shift as it binds the antigen. Okay, so in summary, I think we were able to accomplish what we set out to do, solve these series of different antibody-antigen pairs in sort of an iterative, fairly quick manner. And despite the sort of compositional and conformational heterogeneity that was present in these samples, I think using iterative rounds of heterogeneous refinement, 3D classification, and 3D variability, we were able to sort through that heterogeneity and arrive at a set of particles that allowed us to reach higher resolution. And of course, there's always going to be this trade-off, how much heterogeneity is present in your sample. In our case, I think the level of heterogeneity wasn't too extreme. The data sets that we collected are about one overnight data set's worth of images, and that was enough to start with a particle set that was large enough to trim down to what was needed. But of course, if your sample seems to have a higher degree of heterogeneity, maybe we need to collect a larger amount of data. So yeah, some amount of sample dependence. We also saw that the anti-LC kappa VHH can be a useful tool to stabilize these fabs that have a kappa light chain, not only as a small mass enhancer itself, but to, again, stabilize that hinge in the fab, which, effectively can increase the mass a little more itself. This is something that we do offer for client work now. So if your sample does include an antibody with a kappa light chain, we can try adding this as an additive to see if it helps with sample behavior. And then on the scientific side of things, the epitope mapping series, the series of structures that we were able to solve, revealed some interesting entropic and enthalpic contributions in the form of these side chain mutations that we could see at the interface, or with the loop rearrangements that we could see in OMEGL, and how these different contributions affect antibody antigen binding. If you're interested in the full story, particularly how these structures fit into the context of the mountain of biophysical data that the Philips lab has been generating, the preprint for that should be coming in February, which is soon. So we'll be sending a link to that to the attendees here today if you're interested in following along.
All right, I want to thank you, Kenny, for a really excellent presentation. So we are now going to go into the Q&A portion of our webinar. Just as a reminder, in your toolbar, you'll see a Q&A option. So if you post your questions there, we'll be able to address them live. If there are any questions we do not get to today, we will follow up with you after today's webinar via email. We're going to get right into it. So whenever you are solving a structure, there's always this question of how do you know when to stop versus when to keep trying to push the resolution? So one of the questions is, why wasn't a higher resolution, like below 2 angstroms, pursued in the experimental electron density maps?
Yeah, probably I think the question is asking, because the initial complex structures that we solved, those data were collected on a glacios microscope. So I think, there, of course, is the option to go to the Krios to try to get to higher resolution, or to try to collect additional data to try to get to higher resolution. I think one of our original goals when we started this was we wanted to see and possibly offer sort of a fast turnaround epitope mapping product. And so the quickest way to do that is, because we do a lot of our earlier work on the glacios, that turnaround can be shortened if we stay on the glacios. We just immediately go into data collection, collect the data, process, and try to quickly turn that around. So part of it was that reason. But again, if we have a client project and the client really wants to push the resolution as much as possible, then there are options for that, like collecting more data or going to the Krios to try to push resolution as much as possible.
The next question addresses something that is also on my mind, which is that for the Fab VHH work, this is very impressive that you were able to get to 3 angstrom-ish with such a small protein. So they mention around 60 kilodalton. You'll have to correct us what the exact molecular weight was, Kenny. Could you comment if it's feasible to consistently get structures of comparable resolution for proteins of this size?
Yeah, so I think it was around the 60 to 65 kilodaltons. I mean, at this point, we've been able to do it with the fabs. We have some other structures that we've internally generated around the 50 to 60 kilodalton mark that we've presented on in the past, that we've gotten to a similar resolution. So I think we're at a stage where it's certainly worth a try. Again, it will depend on sample behavior. Like in this case, we were able to stabilize pretty much the entirety of that fab so that we didn't lose our effective mass. But if you have a small protein that also has some flexible regions, that could add to the challenge. So yeah, we're at a point where we maybe won't shy away from it, but it'll be, sample to sample.
In order to get to the final data, there were a couple different tools that you used in CryoSpark. So this question is, of the heterogeneous refinement, 3D classification, and 3D variability, which method gave you the best classification to separate the particles in your experience? Or maybe it was a combination.
Yeah, I think so. The 3D classification tool I've had maybe mixed success with in the past, but for this specific project, I think it did a pretty good job in separating out the classes that have like different numbers of fabs and then, in different RBD conformations. So I think that the 3D classification tool for this project was pretty good in sort of trimming it down initially to the particles that we then took into heterogeneous refinement and 3D variability. That was sort of iterative as to try to see if an additional round would improve resolution further.
Yeah, that makes sense. You touched on some changes to the RBD. So there's a question, is there any conformational change of Omicron RBD upon fab binding?
As in, does the epitope-paratope? So I think some of the pairs, if I'm understanding the question correctly, were solved, especially of the non-VHH ones. I think some were of the RBD in the up and the down versus the down. And I don't think it looked the same in either case.
So this question is about the consistency of results when you're processing something for cryo-EM. The question is, if you were to do the same cryo-EM experiment multiple times with the same sample, using this method, do you expect you would get similar results? Do you expect there would be some variability? I guess how consistent would the results be?
Of the same exact sample 3 times.
Yeah, I think it's asking about the reproducibility of the experiment.
I think if it were the same sample 3 times and we, collected similar size data sets in each case, I think based on previous experience, I would expect, yeah, us to get the same results each time.
And you touched on that when you started the experiment, you were using the 200 kV microscope, and then at some point made the switch to the 300 kV. So, what made you decide to make that switch to the different microscope?
I think it was mostly an add-on project, not necessarily within our epitope mapping project that we had in mind. And in that case, the preliminary work was all done on the 200 kV Glacios microscope, the initial optimization, but we realized early on that to try to push the resolution for that sample to a point where we could potentially build a model, we had to go to the higher kV.
And then getting into something a little technical about the microscope, somebody's asking what's the detector on the microscope that we used?
For the Krios, it's a K3, and the Glacios, it's a Falcon 4.
So this data is, of course, just one part of kind of a bigger project that's being carried out by the Philips lab. I don't know if you can touch on what are this other orthogonal data that maybe the lab has been using to either validate or complement some of these conclusions.
Sure. I don't know all of the biophysical data that they've generated off the top of my head, but I think one of the more interesting results and one of the things that they were more excited about was this isoleucine to tyrosine mutation in the CDR2 loop in the heavy chain. Here, where they were seeing through their biophysical data that mutation appears to occur before other mutations around it that are also important and improve binding affinity, for example. And so I think this structure sheds some light onto why that may be. I think when the isoleucine evolves to this tyrosine, it appears to maybe cause a steric clash with that loop that has to shift downward, and that could be a contributing factor in how that loop reconfigures when it's in its evolved state.
All right, so I'm going to ask our final question here. For this sample, did you notice that the incorporation of a bound fab altered particle behavior, such as causing preferred orientation or resolving it? And would you expect that incorporating a fab would change the behavior in kind of a predictable manner?
The fab or the VHH?
So this one is asking about the fab, but I think if you want to touch on the fab or the VHH.
We didn't look at the spike by itself without any fab. I don't know if removing the fab completely and just looking at the fab, if that would alter its, orientation behavior. For the samples with and without the VHH, we didn't see too much difference in either case. In neither case were the samples exhibiting signs of preferred orientation. But I think it's possible that, adding different fabs could, I think, affect how it interacts with the ice and affect the orientation.
All right, so we are coming up on 2:30. So we are going to conclude today's webinar. We got a lot of really awesome questions. We were not able to answer all of them during the Q&A. So if we did not answer your question, we will follow up with you via email. Just as a reminder, everyone who registered will receive a recording of today's webinar and also a link to the preprint of this paper once it becomes available in the coming weeks. You'll also be receiving updates about any upcoming webinars that we may be having. Kenny, I want to thank you again for a really excellent presentation. Thank you to everybody who joined us today, and we look forward to seeing you next time.
Right. Thank you, everyone.